There were two main experiments that led to the discovery that a specific codon led to a specific amino acid, and that led us to find all of our different codon combinations. In this video, we'll be reviewing both of those experiments and their significance. The first of these experiments that we'll focus on is Nirenberg and Matei's experiment that showed a specific RNA sequence led to a specific amino acid. So they took cell-free matrix, which is just all the parts of the cell taken out of the cell, and they mixed it with a poly-U mRNA chain. So they put that in there, and then they added all the different amino acids into their test tube. So they put the amino acids in there, and they did this a total of 20 times. And each time, one of the amino acids, so in this case it was phenylalanine, all of them would have been radioactive. So the only time that a protein that was detectable actually formed with the poly-U chain was when the phenylalanine was radioactive, and it formed a protein of phenylalanines. They did this again with a poly-A chain 20 times, and the only time it would form was with the lysine. And they also did it with a poly-C chain, which formed a proline-based protein. So this showed that a specific RNA sequence would lead to a specific amino acid being formed. The next experiment was done by Nirenberg and Leader, and this was used to identify the remaining 61 codons. They did this by taking trinucleotide mRNA sequences, also known as codons, and they would mix it in a test tube with ribosomes and with different tRNAs that were attached to amino acids. And then every time they would do this, they did it over and over and over again, they would take a radioactively marked amino acid that was attached to a tRNA, and they would put it in there, and then they would filter out the product. And the tRNAs and the mRNAs would just filter through perfectly fine, but if they attached to a ribosome, then they would not filter, and they would be able to identify based off the radioactivity which codons paired with which amino acids. And this is how they identified the remaining 61. These experiments by Nirenberg, Leader, and Matei are incredibly significant to the field of biology. They showed how this is an unambiguous process, which just means that a specific codon always encodes for a specific amino acid, never multiple different amino acids, and how this is degenerate, which just means that you can usually predict what amino acid will be formed based off of the first two nucleotides in the codon. This is incredibly significant because it really is what developed our understanding of how our genetic material is used to form these primary structures of proteins. And this is also a nearly universal code, so this applies to almost every organism on this earth.